quite a while trying to convince a crowd that what I'm talking about isn't a bunch of BS, but now I have to try extra hard. Let me just call up my talk here. Okay, well, we're good. So before I talk about what I'm going to talk about tonight, I do want to say some thanks. I would like to say thanks to everybody who decided to invite me. I'm sure Lorne and Louise Trottier had a say, and I want to thank you for not just tonight, but for being very generous about informing the world about what's fact and what isn't fact, because we live in a world that is increasingly hard to distinguish between those two. So can I just have everyone thank them again, because it's really important. Okay. And this is very personal to me, because I'm faced with this problem every day. If you were to type in my name into the internet, you wouldn't most likely find my book, or you wouldn't most likely find my research. You'd most likely find products sold by people who I have nothing to do with, who are using made-up quotes from me with my name all over the internet. And I cannot tell you how much money I've spent trying to stop that, and it's impossible to stop it. There's even a company called Sinclair Labs that sells products based on my lab's research. And I just found out tonight there's another copy of my book, a pseudo book. There was one on the internet. So my book is called Lifespan. And this was 10 years of work into this book, and I poured everything I had into it. And within a few days of being on Amazon, first of all, I was very lucky. It shot up to being a New York Times bestseller, so that was great. And then I'm looking online to see how the reviews are looking, and I see that there's a book that looks great. It's getting some good reviews, and it's written by Daniel Sinclair, and it's a rip-off of what I've written. And there are people writing to me saying, "How do you? I can't. This book sucks. I've gotten this copy." And then I learned tonight there's another one out there. So please look for the book that has my name on it and is actually a PhD. That would be good. I will be writing to Mr. Bezos actually after this to tell him to help me shut down these things. But we live in a world where it's very easy now to rip things off and take people for a ride. And we see that not just on the internet, but we see it in cosmetics. We see it also in other parts of the world, in drugs as well. You can't even trust what you're going to get. And I can tell you, and I'm going to tell you, I wake up every morning to more than a few hundred messages and questions from people asking me, Dr. Sinclair, what can I trust? And I just can't. I could spend my whole day replying to all of them. But tonight I'm going to tell you about some of what I think you can trust, and that's the science. And all my disclaimers down there. I work with biotech companies. I've started 15 of them. I have no affiliation to supplement companies, so everything I'm going to tell you about products that are out there, I have nothing to gain, as Dr. Schwartz also said. Okay. So these are two people near and dear to me. The lady on the left is my grandmother, and she is a lady who raised me when I was young. She gave birth to the person on the right, who is my father, obviously, when she was 15. It was a bit of a scandal back in the 1930s, 1939, and so I had a very young grandmother, and she taught me that, having lived through World War II and the aftermath in Hungary, that humans are capable of great bad, and we are going to hear Dr. Ruth Westheimer talk about that. She understands that better than I do, but also she also realized that children are innocent and they're the ones that you should trust. And so she told me to stay young. She told me 6 was the best age. Now, and she used to read A.A. Milne all the time to me. And there is a stanza in Winnie the Pooh that says, "Now we are 6. I'm going to stay 6 forever and ever." Now, I cannot stay 6 forever and ever, nor can you. But could we one day go back to being 56 or 46 or even 26? I'm going to tell you today that That's not out of the realm of physics, nor is it out of the realm of biology. And the other reason I want to show you this slide is that my grandmother, Vera, she didn't like to be called grandmother or Nana. She just wants to be called by her first name, which was radical in the 1970s. She told me that she didn't ever want to grow old, but I saw that happen to her, and we've all had people in our family who have spent the last decade or so of their lives in a state that you wouldn't wish on your worst enemy, and that's what happened to her. She went from being the most vivacious, funny, outrageous, rebellious person. She was kicked off Bondi Beach for wearing a bikini in the 50s. She went and lived in New Guinea for 2 years as a woman in the 1960s. She went from that to somebody who just gave up on life, didn't go out of the house for 10 years or more, and said, David, that's just the way it goes. And I refuse to believe that that's the way it goes. We've never, as a species, said that's the way it goes. We didn't say that about infections, we didn't say it about cancer, and we shouldn't say it about aging either. So she had a terrible end to her life, and this is fairly typical the way we do medicine these days. We treat one disease at a time, but we don't fully treat the whole body. And for many people, their heart keeps beating, but their brain does not do well. And what I'm hoping to do with my research and with my colleagues is to keep the whole body young, not by creating a miracle pill, as Dr. Schwartz has told you, some people claim there is one, but by learning how does the body fight disease? How is it that there are species that live for 200 and 300 and 1,000 years? How do they do it? Some of those animals are very closely related to us, such as the bowhead whale. And if they can have a workaround to aging, so can we. And my hope is that my father and my generation, and certainly our children and their children's children, will live lives that are very different to us, the way we look at people who grew up in the late 1800s, early 20th century, where it was common to die from childbirth or an infection from a splinter, and there's no way we'd all go back and live in those days. Okay, so this is my picture of aging. This person, I'm told, is from Australia, where I'm from, where we're all beaten down by the sun. Clearly, this person has had more sun on one side of their face than the other. But this is the face of aging. This is what happens. But this doesn't just happen to the skin, it happens to all parts of the body. And the question is why? And this is such a simple question, but I have to say it again. Why do we age? Does anyone spend much time thinking about that? I know a lot of researchers don't, and I certainly know a lot of doctors don't. But I've thought about this since my grandmother told me she was going to die one day, and so would all my friends and family and myself. So that's a shock that we all go through when we're about 4 or 5. But we forget about it. We actually have a part of our brain that helps us forget about that because it's too tragic. And often we think about it until it's much too late. But I'm going to tell you tonight that if you think about it and you take action, there are things that you can do right now that actually, we believe, can delay aging and help you reach the age where Dr. Ruth, she's 91 and as fit as a fiddle. I want that for everybody in the world. I don't want us to all live to 200. That's not the goal. It's to be healthy until much later. And if we have a world like that, it'll be a much better place, the same way anyone from the 1800s would look at today, and this would be utopia to them. Now, we can talk about all the issues that we'll have to overcome society-wise, but actually the savings are in the tens of trillions of dollars globally, and that's money that I think can solve any problem. All right, so speaking of pseudoscience, we went through the 1970s, 1980s, and we still had a hangover from the Manhattan Project, where we believed that DNA damage and radiation was the biggest problem, and mutations in our body. And I think if most of us go to a party and somebody starts talking about aging, they will say, DNA damage causes aging, or mutations cause aging, and you need to drink a lot of antioxidants. Drink Pom Wonderful and you'll be good. I have some bad news for all of you. Even though there's $200 billion spent on antioxidants a year in North America, in drinks and food, I don't think they do a lot. I'm sorry to say the science doesn't back that up. Now, they're not useless. They will do some good, but they're not going to make you live longer. They've had a huge failure over the last 30, 40 years of tests in terms of slowing down aging. So if that's true, what are we going to do? Well, over the last 10 years or so, scientists like myself have come up with a list of what we think are the main causes of aging. And we call these the hallmarks of aging. And you've probably heard of some of these. They're in the news. Telomere shortening, the ends of chromosomes, mitochondria, the energy packs die out. We have senescent cells, which are zombie cells that accumulate. We have diabetes, deregulated nutrient sensing. We have a loss of stem cells. There's a lot of people trying to replace stem cells. And we have what's called epigenetic alterations. And this is great. About 10 years ago, we said, as a field, we've finished. Eureka. It's a beautiful diagram. Are we done now? We've figured out aging. But I wasn't satisfied because that doesn't explain, that beautiful wheel doesn't explain why they occur in the first place. And just saying bad stuff happens is not a good enough explanation. In the same way that we say, oh, things drop to the floor, that's how it's always been, but you still need to explain why they drop to the floor. And so we do believe that if we address each one of those 8 or so hallmarks of aging, we will be healthier and live longer, no question. But we're still building 8 dams on 8 tributaries. Is there an upstream cause of most, if not all of them? And I believe that there is. Now, scientific paradigm shifts come around once every decade or so, and we're in one of those right now in the aging field. Many of my colleagues have tried to test this theory of mutation causing aging, the mutation theory of aging. And what they found is that if you mutate an animal, it can live a normal lifespan. We've had mice in the lab that have huge amounts of mutations in their nucleus, in their chromosomes. They don't age. That's weird, right, if that theory is true? The other issue is that if you look in our bodies, there's surprisingly few mutations. Now, you can find them, of course. They're there, and the more sun damage you get and the more exposure to a variety of toxins, you'll have more. But they're just not in the abundance that you would expect if this was the primary driver of aging. So we're going through this paradigm shift, and this always happens when experiment doesn't match theory. And it's a very tough time for scientists because there's a lot of fighting, there's uncertainty. It's a period of chaos. The models drift, we try to move the models to fit the data, but then eventually we come up with a new theory that can explain all of this new data and all of the past few hundred years of research. And so what I'm going to propose to you tonight is what I think is the main driver of aging, why we don't stay young forever. And it can be put the following way. Aging is simply a loss of information. Okay? So we live in an information age. So we know a lot about information loss. We have information loss when we send out radio signals. We have a lot of noise. You can hear it as static. So what kind of information am I talking about here in our bodies? Well, the one we always hear about is the genetic information. This is, of course, what we inherit from our parents. It's the DNA. It's our digital information. And instead of being zeros and ones, it's A, T, C, G, encoded as DNA. And digital information is really robust. That's why it's been the go-to molecule for life for the past 4 billion years. I was shocked when I first got to the lab and found that you could boil DNA and it would survive. I thought this would be the most fragile molecule of all. And in fact, we can now pull it out of fossils and preserved remains, many, at least tens of thousands and hundreds of thousands of years old. So digital information in the form of DNA is very robust. And as I've mentioned to you, it seems to last a lot longer than we thought, even within our lifetimes. So if it's not DNA, what is it? So the other type of information that's just as important, and without it we would be dead in seconds, is epigenetic information. And what I mean by epigenetic information is how the cell controls its genome. So the DNA in our bodies is not just flailing around like it's a piece of string in a swimming pool. It has to be regulated very precisely. The reason is that every cell has a different function, or at least every cell is part of a tissue that have very specific functions. The brain cells, there are hundreds of types in our brain. Each type has to have its own identity, very different from a skin cell or a liver cell. But they all have the same DNA. So what makes them different is how their DNA is packaged. And DNA that's wrapped up very tightly, like you would spool a hose on a driveway, that's chromatin, we call it chromatin that's off. And genes that need to be switched on, nerve-specific genes in the brain, are looped out, spooled out in these big loops so the cell can now read them and make proteins out of it. And that information, unfortunately, is predominantly analog information. And I think most of us here are old enough to know how much analog information sucks. Anyone who's had a cassette player or a record player, or as Joe Biden would say, a phonograph, we know that these things are really bad at storing information long term. In fact, if you try to play an old cassette tape, it's probably not going to play too well, let alone if you've scratched a record. And even a CD player, a CD will get scratched as well. You know, I have to teach medical students at Harvard Medical School and explain to them these days what a CD is. I have to say that it's this thing we used to put music on it. It could hold up to 10 songs. It was incredible. So the fact that the epigenome is analog, and the reason it's analog, by the way, is that it needs to be. It needs to respond very quickly to the environment. It's what changes when you eat a meal. It's what changes when you get hit by damage. Say you have an X-ray and you damage your DNA. It's the epigenome that responds and changes how the genes are switched on and off. But the important part of aging is that once a cell responds to a threat and starts changing the way genes are turned on and off, I believe it doesn't always reset its original state. And over time, we lose our epigenetic information. Those green things, by the way, we call those histones. Those are proteins that are very useful for bundling up the DNA. And if you pulled out a piece of DNA from the cell, it'd be about 6 feet long. But those histones would be a lot like holiday lights strung along. And they're actually largely what controls which genes are on and off. Now, there's another modification that changes genes on and off, and it's called DNA methylation. And Dr. Schwartz wouldn't tell you, as a chemist, that a methyl is just a hydrogen with carbon with 3 hydrogens on there. And what we found out is that these methyls, they don't just control what type of cell a cell will be, but they also accumulate in specific ways as we get older, which will be important in a moment. But when I talk about epigenetic noise, not noise from a radio signal, but true epigenetic noise, what I'm talking about is a cell that starts out beautifully young, functioning 100% when we're born, usually, and even as we're teenagers, things work pretty well. But over time, we're still aging. And what's changing is the organization of our DNA, the analog part of our genomes. And so in this diagram, you can see that genes that were once on, this red dot here with the RNA coming out, that's a gene that's on. It may eventually be shut off over time by accident and vice versa. And those methyl chemical groups will start to get messed up over time. And we can read that. And we can actually tell you how old you are by reading those changes with those chemicals on the DNA. It's called the epigenetic clock. And what's scary about that, and I need to give a shout out to my colleague Steve Horvath at UCLA who helped develop this clock. I could take a blood sample from each of you and very precisely predict when you're going to die. It's that accurate. And if you smoke, and if you don't exercise, and if you eat 3 full big meals a day, your clock will go faster. We now know that. But there's some good news. I'll get to that in a minute. But the best analogy I can give you tonight is that our cells are like compact discs that get scratched. The information is still there, but the cell doesn't read that information. So what we've been looking for is the polish. How do you get back the information? The other way to think of it is that our epigenome is the software of our computer. And then a third way, one that people are telling me from in the book they like it, is that our genome is the piano, but instead of having the normal set of keys, you have 20,000 keys. And the epigenome is the piano player. And over time, she starts to lose her eyesight. She can't play the notes. And initially, you don't notice if she plays a few notes that are wrong. That's your teenage years. But over time, as you get older, it's a cacophony, and you would just walk out on the concert. But what we are trying to do is to find a way to tell that pianist how to read the notes again, give her glasses so she can see again, or just get rid of her and get a new pianist. All right, so I've jumped from a CD to an island. Just very briefly, we think that life was set up to be this way because it's very important to coordinate your epigenome with DNA damage. And this is an example of what the early Earth looked like 4 billion years ago, we think. And it was full of noxious gases, and that there were early life forms living on there. But there were life forms that had a very particular gene survival circuit that was composed of only 2 genes. One that controlled how genes are turned on and off, and then a gene that makes a protein that repairs DNA. And the reason for that is you don't want to always be growing because sometimes times are tough. When you're hit by cosmic rays or you run out of food, you shouldn't keep dividing and producing offspring. You want to hunker down, and those are the life forms that survived on the early Earth. And we are the descendants of those cells. And we carry that same genetic survival circuit in our cells today. Without that survival circuit, we couldn't repair DNA, and we couldn't survive. But if my theory is correct, that is also the reason that we age. So this is my representation of a chromosome here. And these balls here, these yellow and blue balls, represent proteins that control genes. These are epigenetic regulators. And the ones that we work on, that I'll tell you about later, are called sirtuins. But there are many of them in the cell. But it's the principle I want to get across tonight. What we've discovered in things as simple as yeast cells, and even true in mammals, is that when we damage a piece of DNA, if we get in the sun too much, like a stupid young Australian kid that I was, or we have too many x-rays, or, God forbid, you have to undergo chemotherapy, you're going to be damaging the DNA. And in response, you'll see in this diagram, these proteins will have to move to help repair it. That's part of the survival circuit that helps turn on the response. But over time, if you keep repeating this pattern, you will lose the original pattern, like playing a tennis game and losing the tennis balls out of the court. Now, this is a nice theory. We had a fair amount of evidence for this over the last 25 years, but we didn't have proof, or at least a really good test of cause and effect. And that's what we like to do as scientists, if we're really going to test a theory rigorously. So what we decided to do was basically that, but in a living organism. We've done it in cells, we've done it in human cells, we've done it in yeast. But what if we took an animal and did that? If the theory's correct, right, we should get epigenetic noise, and we should get aging. Now, it took us 10 years, and our publication just came out online, if you'd like to Google it. And I'm going to tell you about that tonight. Now, we engineered a mouse, and hats off to the mice. Thank you for the sacrifice for the greater good. We engineered a strain of mice where we could cut their chromosome but not cause mutations and distract those proteins and get them to move around. And if we're right, those mice should get old in every way. And that clock that you can measure should advance faster. Now, if we're wrong, and there's 99.9% chance we're wrong, the mice would die, they'd get cancer, or nothing would happen. And most people said that would be the case. But what we got was the following. We scratched the CD in these mice, and they got old. These mice are exactly the same age, with identical genomes. They haven't got any difference in terms of mutations. They're mutating at the same rate. But when they were young, we scratched the CD of one of them, the one on the right. So what did we actually do? We put in a gene from a slime mold that we could get, and we turned it on for 3 weeks when the mouse was young. The mouse didn't notice. It was quite happy. It was as though, you get an X-ray, you don't feel that. But then, because we disrupted its epigenome, its cells started to age. They started to lose their identity. What made those nerve cells in that mouse's brain started to go away. And we find that the mouse on the right, its cells have largely lost their identity. They're barely functioning, and they're becoming like a melange of a variety of cells. So let me put it this way. Aging, we believe, is the loss of cellular identity. The question, though, is, by the way, what that ends up is, if it goes too far, is senescence. And those cells hunker down, they stop dividing, and then they wreak havoc, including inflammation. But then the question is, are they truly old? And as I mentioned, we can measure their age, and this is based on those chemicals that naturally accumulate, the one that we can do in our bodies or in mice. In this case, we've measured their blood biological age, not their birthday candles, but their actual biological age. And the red dots represent that mouse on the top, and it's 50% older. And if you look at its organs, the organs are also older. And if you look at its biochemistry is older, and they live shorter. And they develop all the signs of aging. In fact, we can dial this up. We can accelerate aging quite severely in these mice. We can make them age within a matter of months. And what we're excited about is that these mice could serve as a model for human aging and human diseases. And we've had very poor success in treating Alzheimer's disease. And I would argue the reason is we're testing our drugs on mice that are only 6 months old. How can you expect that to yield important results? But these mice, we can age them 80 years within a matter of months if we want to. We just feed them a drug called doxycycline, and then it turns it on. So on the left, you can see on this slide, are these chemical groups, these methyls, which are in gray. And there are enzymes called TETs that remove these methyls. That'll be important in a minute. But what we're asking, in my lab we're asking over the last 2 years, was can we take an old mouse and remove those chemicals in the right way to make that clock young again? Now, we don't want to strip all the methyls off. You might say, well, don't you just turn on those TETs and get rid of those methyls? Well, if we do that, you know what we get? We get the world's biggest tumor because the cells would just grow like a cancer. What we needed to do was to get back the pianist that can play just the right notes at the right time, at the right place. That's pretty hard to do, a lot harder than stripping off these methyls. Now, stripping off the methyls is not stupid. As you'll see, it's a very important part of biology now. Now, one of the important things that you need to know is that adult neurons, after we're born, they don't regrow. But if we're very young, or if, say, we're still in the uterus, if we damage our optic nerve or our spine, it can grow back. And there are some species that can grow back entire arms or tails. And there are jellyfish that can go from a few cells and respawn an entire new individual. So aging and new body parts can be regrown and reset. But our nerves are the most pathetic cells in the body. You damage them once and they're never coming back. So we like a challenge in my lab, and a student in my lab, Wancheng Lu, decided that perhaps we could try to reprogram neurons to be young again by tickling those TET enzymes to get back the pianist. And what we expected to see was that if we were right, we should be able to regrow nerves in the animal instead of having them sit around and just vegetate. All right, so this is Wancheng Lu on the right. He's just a young 20-something-year-old guy who grabbed his PhD by the horns and went for it. The man to his left is Shinya Yamanaka, who won the Nobel Prize in 2012. This photo was taken actually yesterday. They were hanging out at Harvard. And this is a dream come true for Wancheng because his research, our research, stands on the shoulder of this guy. So if you don't know Yamanaka, he found that there was a combination of genes, 4 genes called Yamanaka genes, that if you put them into adult cells, let's say one of your skin cells, you can reprogram those cells completely to be a stem cell and then coax those cells to be just about any other cell type you want. You can make sperm, you can make egg, you can make both, you can fertilize them if you want. A remarkable discovery and well-deserving of a Nobel Prize. But we decided that putting all 4 Yamanaka factors into an animal would not be a good thing. As I mentioned, you'd get the world's biggest tumor. So just a little bit about Yamanaka factors. They're called O, S, K, and M for short. OCT4, SOX2, KLF4, and MYC. These are what's called transcription factors that control other genes, including those TET enzymes that scrub the DNA of those methyls. But what we didn't know was, is it possible to partially reprogram a cell, not to be primordial and stem-like, but just enough safely to be young again, to polish that CD and play the music of our youth once again? And the brilliant breakthrough that came from Wancheng was that he said, you know, there's a problem here. One of those genes is an oncogene, meaning it can cause cancer. So he left off that gene at the end, MYC, and he just built a virus that contained O, S, and K, packaged it up, and injected it into a mouse to see what would happen. Now, we had some idea that this might work because a fellow called, a professor, I should say, Juan Carlos Belmonte, a good friend of mine at the Salk Institute, a few years ago had shown that if you turn on all 4 of these genes in a mouse, a short-lived mouse, it would die within 2 days. Now, that's not going to make headlines, and it's not going to be an FDA-approved therapy anytime soon. But what he did do that was very clever was he turned those genes off again after 2 days, and the mice recovered from their stress. And he kept doing that for the rest of their life. Now, that's a horrible treatment to a mouse, but those mice lived 40% longer. But everybody that I talked to said, that's just an artifact. If you stress a mouse and almost kill it every 2 days, that's what's going to happen. But we believed in it, and what Wancheng did is quite remarkable. Now, we have collaborators at Harvard, and I really owe them a great debt. We're not expert at optic nerve crush, but the lab that we collaborated with is, and what they did was they pinched the back of an optic nerve of a mouse, and that's the best model to understand how to regrow nerves, which one day may be used either to regrow the eye or to fix spinal cord or other injuries. But right now, science is at a stage where you can barely grow back any nerves at all. So what you can see up here, hopefully, is that this is a nerve that was pinched across here, and a lot of these nerves where it's orange stain died off. This should be bright orange. And these are the ends of the nerves that run that way, towards me. They're heading towards the brain, and they've all died off because of the pinch after about 2 weeks. Now, we did the same experiment, but we introduced with a virus, a gene therapy that we could turn on with an antibiotic, doxycycline, and we fed the mice doxycycline and had a look what happened. And what we saw was quite remarkable, that the nerves started growing back towards the brain. Somehow they knew where to grow. And if we leave this longer, they grow all the way back to the brain, which is quite a distance. And we know that if we get rid of those TET genes, those methyl erase genes, part of the epigenetic clock, this doesn't work. That's pretty interesting. Now, how do you tell if a mouse can regain eyesight? Now, in this experiment, what we're doing is we're not crushing the eye. We did a couple of things. We gave mice glaucoma, pressure-induced damage to the retina, and that was their vision was largely restored by a virus, at least 50%, which is unheard of. Glaucoma you can't typically cure. Now, these are old mice. These are 1-year-old mice, and these mice lose their vision at 1 year of age. And ignore the feces, that's irrelevant. They're just a bit scared, standing on a platform. But what Bruce Cassandra did, our colleague, was he showed the mice a moving line, and if mice can see, they will move their head. By the way, so will we. It's called the optomotor response. Now, this old mouse has not been treated, so you wouldn't expect it to be able to see. We can also, by the way, put electrodes back there and see if we're just fooling ourselves, but we do that too. But now, let me see if I can get the next slide going. So this is an old mouse that has been treated for a few weeks with our gene therapy to reprogram the retina to be young again. And we find that it can see just as well as a young mouse again. We've completely restored its vision. So what that tells us is parts of the body, particularly the retina, are probably not degenerate. They just forgot that they were optic nerves, and you can reprogram them to work again. which is stunning. What that means is there's a backup copy of youth in all of our cells, if we're right. And it also means that this DNA methylation clock, this epigenetic clock that we can measure, may not just be a measure of age, but it might actually be part of the system. It's as crazy as the following, that if you have a clock on the wall and you move the hands backwards, we would say that doesn't change the actual time. It doesn't move time backwards. But what this is saying is that maybe it does change time. It really does change our biological age. And so we hope to do tests in glaucoma patients in about 2 years with gene therapy in the eye. But we're also going as fast as we can to identify simpler ways to reprogram parts of the body. We haven't found any safety issues. We've had this gene therapy delivered in the veins of mice for over a year, and they're doing perfectly fine. So we're not causing tumors as far as we know, and we're optimistic that this could be a new way to rejuvenate the body and reset the clock. Now, we don't know how many times you can reset the clock, actually. We've done it once. But what if you could reset the body 100 times, 1,000 times? That's when things get really interesting. But you might be saying, oh, David, this is going to cost a lot of money, and it's not going to be around for another few years, and even then, I don't know if I can get it. So what can I do now? Well, if there's one word that you might want to take home with you, it's this one: hormesis. It basically means anything that doesn't kill you will make you stronger and longer lived. We cannot let our bodies become complacent. And it all goes back to that original survival circuit in the early life on the planet. It responded to stress. And if we don't activate it, it just sits there and it doesn't defend the body. So you want to tickle it. But of course, if you step on a snail, it's not going to live longer. It's just going to die. What we want to do is to tickle it. So how do we do that? We give it a little bit of stress. A little bit is good, a lot of it is bad. And there are factors that we've discovered, such as mTOR and AMPK, and the ones that I work on called sirtuins, that are in all of our bodies that are activated by a bit of hormesis. And there are other ways of activating them, such as with drugs like rapamycin, which is an immunosuppressant, so I don't recommend that. Metformin, I'll talk about in a bit. And I work on these molecules here, what are called NAD boosters. And there's also resveratrol, which Dr. Schwartz talked about, which was just a proof of concept molecule. We have much better molecules now. So if there's one thing I can tell you in your daily lives to do, after reading thousands of scientific papers and researching a lot of animals and humans, it would be this: eat less often. Not starvation, not malnutrition, not at all. But 3 meals a day with snacks in between, never feeling hungry our whole lives, that's ludicrous. And I don't even know why nutritionists ever recommended that. Naturally, we're supposed to be hungry. And without that, our inbuilt defenses that I just showed you on the slide remain relatively inactive. Our bodies don't fight disease, they don't fight aging. So eat less. What I do, and I don't tell anybody what to do, but what I do is I skip breakfast as best I can, and I eat a late lunch, and sometimes I go all the way to dinner. That works for me. Some people skip 2 or 3 days, sometimes a week. But please, whatever you do, don't always be full. It's craziness, and no wonder we have an obesity epidemic with that attitude. Get up and move. Now, this is not new. You don't need someone from my lab to come and tell you this. But what we've learned is that the reason exercise works so well is because it's activating these longevity genes. And with small molecules that we found, such as these NAD boosters and resveratrol, we can actually mimic the benefits of exercise. And when you combine them with exercise, we think you can have an even greater health benefit. But move. Go for a walk. If you're able to do it, lose your breath. Run a little bit. But really, you've got to move and maintain that body strength as well. Lift some weights, stretch, because as Dr. Ruth said earlier today, the most important thing to living a long life is hanging on to the handrail or to good-looking men. They both work. I also looked at other types of hormesis. This is the sauna. I was skeptical that this was true. Of course, heating your body, that's craziness. If we heat yeast cells, they live longer, but a human body, that's craziness. But actually, the data is interesting. I think it's compelling that a number of studies in Scandinavia have found, looking at thousands of mostly businessmen, caveat, that they are protected against heart disease. So I think that that may work, and if nothing else, you'll feel good in the middle of winter. Cold baths. Now, this also I thought was unlikely to work or do anything. But actually, even in my field, these sirtuin defenses are activated by being cold. There's one in particular called SIRT3. There are 7 in total. SIRT3 comes on when you're cold and helps cells survive and also increases their metabolism. And it gets activated by building brown fat, which can be coaxed into existence and proliferation, typically on your shoulder blades. And we didn't even know this kind of brown fat was there until about a decade ago. So these days, I do that. I jump in a cold pool with my son every weekend after being in the sauna. And at the very least, I feel quite lucky to still be alive. So there's this plant here. It's the French lilac, or goat's rue. It's a less popular name. And it produces a molecule called guanidine. And these guanines actually were found to be quite useful for treating diabetes, type 2 diabetes. And then scientists, actually, chemists, went to work and created better versions of this. There's a biguanide called metformin, which is the frontline therapy across the world for type 2 diabetes or high blood sugar. And actually, the World Health Organization has declared it an essential medicine for humanity. So what does metformin do? Well, it certainly helps the liver, and it helps your body take up glucose from the bloodstream. That's why it's used as a diabetes treatment. But it's also been found with my colleagues and I to extend a mouse's lifespan. And this pathway, remember I mentioned AMPK, that's one of the 3 main pathways for longevity that we've all discovered. And it has multi-actions. It works on fat, it works on the immune system. And studies of now 100,000+ people around the world who've taken metformin indicate pretty strongly, in my opinion, to protect against many diseases of aging, from cancer to heart disease, even frailty and cancer. So this is really, I think, the closest we have to evidence that there is a drug already out there that can slow aging. And there are many of my colleagues taking this molecule, myself included. Resveratrol, I won't belabor the point. It's all over the news, has been. If you like red wine and you needed an excuse to drink, you're welcome. It's not bad for you in small doses, but the doses that I take are the equivalent every morning to drinking 500 to 1,000 glasses of red wine, and I do not recommend that. So don't do that. But resveratrol seems to be a pretty interesting molecule because what it does is it activates one of these sirtuin enzymes, and we've discovered that it does this. And we can actually stop the enzyme from doing this in a mouse. And we've shown, though we haven't published yet, that if a mouse can't do this action, resveratrol will not make it live longer anymore. Pretty much proving that our ideas are correct. Now, there are drugs that have been developed, or at least are being developed, that are better than resveratrol, and those have been into phase 2 studies showing efficacy in inflammation, but they remain to be marketed. NAD boosters, if anyone here has been on the internet recently or Googled my name, this will come up. There are a few NAD boosters. You can certainly read more about it if you'd like to pick up a copy of my book. I don't want to go on and on about it. But what's interesting about NAD is that sirtuin enzymes that I just showed you don't work without NAD. And the levels of NAD, we think, go down as we get older. And so NAD boosters get you your youthful levels of NAD and we think fight back. So some of the evidence comes from mice. These are 20-month-olds mice, sorry about the black mice on a black treadmill, but one of these mice clearly is fitter than the other one, and it's the one that's been drinking NMN in its water. And what we traced this to was that the SIRT1 enzyme, the sirtuin, in the lining of the blood vessels was responsible, and it was mimicking the effects of exercise. And that's why these untrained mice now ran multi-marathons, and in fact ran so far that the machine stopped running. And we had to rewrite the software because the software didn't anticipate mice running over 3 kilometers. Okay, so we stand at the precipice of an interesting world where what I've told you today is that it's feasible that we will not just slow aging, but we will reverse the aging process. And this is an example. Now, I chose Bill Murray just for the heck of it. It could be anybody, but imagine a world where you get gene therapy when you're young, and these viruses sit in your body lying in wait, they're not on, but if you have an accident, you cut yourself, you break your spine, you damage your eye, you'll get an IV of an antibiotic that will turn on the reprogramming, will make you into basically an axolotl and salamander, regrow what you need to regrow. You'll go back to be young. We don't know how young yet. And then you go off the antibiotic. And perhaps we could keep resetting the body every decade or so. That's a possible future that our children or our great-grandchildren may actually see. Now remember my father, who was born in 1939. Now he's now 80, and he was not looking forward to his old age. My mother died from lung cancer. She lived a terrible life as well. His mother, as I mentioned, died a very terrible life. We have heart disease riddled in our Ashkenazi Jew family. And I also have terrible genes, massive cholesterol. So my father surviving to 80 is a world's first, as far as we know, in our family. But what he gives us is hope that we can intervene. So my father is on essentially the same program that I am. It does involve the things I've told you today and a few other tweaks, which if you want to read on page 302, you're welcome to. I wrote all those down. I know people would be interested. And also write to me if you'd like. But my father is a beacon of hope, as is Dr. Ruth, that we don't have to accept aging. We don't have to accept just the way it goes. The same way we never accepted cancer or heart disease as just the way it goes. And just this August, my father, who I'm showing you here in the middle picture, we went to Africa together, and he climbed the mountains in Uganda to visit the gorillas. And we explored the great journey of humanity from fossils all the way up the Great Rift Valley. And I'll tell you that was when I realized what I was working on was that important. It becomes very personal, actually, when it's part of your family. And with the smile on my father's face, you can see here, he's gone from a grumpy guy to a guy that just loves life. He just ordered his dream car on a wait list at 80. This is not the kind of guy that you'd think would be old. But he has no aches or pains. He started a new career a few years ago and is looking forward to meeting his great-grandkids. And that's a life I want us all to be able to look forward to. It'll be a world where we'll have a lot less expenditure for healthcare, and the wisdom of our elders will be admired and absorbed by our youth that really need our advice as this world becomes more complicated and scary for them. And I want to say thank you all for spending your night with me and the Trottiers for making this all possible. Thank you.